sometimes called the neural layer, because this is the sensory epithelium. These are the photoreceptors, or rods and cones. And remember, they have their little heads buried in the pigment. But most surprising, perhaps, is that anterior to those photoreceptors are several layers of cells, several layers of neurons. These are neuronal cells that are talking to one another, sort of exchanging information, and beginning the process of processing the visual information. You might ask yourself, what type of information is conveyed from the retina to the brain as it leaves the eye via the optic nerve? And our best guess is that what all these cells are doing is that they are simply conveying their edge detector, basically. If you've ever played around with a photo imaging program, you can actually take a photograph, and all it will, there are settings where you can have it just show the edges of all the objects, and that seems to be in part what the retina is doing. It's an edge detector, it's a shape detector, it's a color detector, and that's about it. Everything else has to be processed. All of the visual information has to be processed in the brain. But as I was saying, there's several layers of cells, and then most anterior is this ganglion cell layer, and this is primarily a series of ganglion cells and their attached axons. And then those axons are going to send their branches, the ganglion cells are going to send their axons to the optic nerve, where they will then be conducted to the optic tracts and chiasm and so on. So you have another image of the same thing here. We can see a sort of micrograph on the left here, and then a pictographic representation on the right. So these are the photoreceptors. Everything up in here is the photoreceptor. This is the epithelium here. I mean, this is the pigmented region up in here. So all of this is just photoreceptors, all in here. And then you have several layers of cells, and their associated axons and dendrites and cross. And some of these, it's important to realize that some of these cells are sending information laterally, and some of them are sending information more vertically in this illustration. But ultimately, when all that processing is done, then once again, the ganglion cells will send their axons off to the optic nerve. Anybody see a problem with this arrangement? What do you notice again down here? The light is coming in this way.
Yeah, that is a problem.
It's crazy.
It is.
So the light goes in, it's the photoreceptors in the back, and the signal is sent back anteriorly to those
No,
cells.
so, well, make sure I understand where you're pointing here. So the light is coming in this way. The photoreceptors respond to the light in some magical, mysterious fashion.
Posterior up there, right?
Exactly.
And then it sends it back anteriorly.
Yeah.
Oh, wow.
Yeah, I mean, would you design things this way? Now, there is one little exception to this general premise. At that fovea, at that macular region, notice what tends to happen. There is a much greater access of the photoreceptors to the direct light. However, even in the fovea, there is considerable occlusion. There's because of all the wiring and all the cell layers. There is significant obstruction. It's just that it's much less. It's much less than other regions of the retina. And so once again, you see a somewhat similar representation. How on earth is that possible? And we haven't even talked about the blood vessels. And when the optometrist looks at the back of your eye there and is looking at the blood vessels, I mean, it's just inconceivable. Sort of another pictographic representation of the light coming through down here to the rods and cones. The rods and cones are at the bottom here. This is the anterior surface of the retina up here. Now, there are some theories about how this works. One of the current theories, and I'm not sure it's been absolutely established, relates to this thing, which is sometimes called a Muller's cell. I'm not sure if it's spelled correctly up there, but then I suppose that's possible. It's usually two L's, but called a Mueller's cell. And you, by the way, you can see these other cells here. You can see the photoreceptors. Then we have the horizontal cells. You can see how they get their name. These are very familiar to you, bipolar cells. In fact, most of the cells along the optic tract are functionally bipolar, even if we don't call them that. They have that sort of shape. And then the amacrine cells and ganglion cells. And what I would ask, I'm not going to ask you to be able to, you don't have to pick these cells out in a picture, but I do want you to recognize them. So when you see the word amacrine cell or horizontal cell, or you should be able to acknowledge that the term bipolar cell also refers to a specific cell in the retina. So just be familiar with the terms that are used to describe the retinal cells. Again, I'm not so concerned that you pay attention to the various plexiform layers, but do know the relationship, for example, between the pigmented epithelium and the photoreceptors, and then the ganglion cell layer up there. I'm sure you understand that that is most anterior. Now, I was talking about this Mueller cell. When I was a child, you could go into a Radio Shack. Remember those? Go into a Radio Shack, and they would have, especially at Christmas time, they would have these remarkable little objects on the shelves that would have long pieces of plastic, long tubes of plastic that would change color. Anybody ever seen these? What I'm talking about? Remember, I don't know what term they used. They used to be called fiber optic, and now is the basis of a lot of the communications for digital traffic. Fiber optics isn't used as a bauble anymore. It's actually used much more importantly. You probably have fiber optics in your car anyway, where it's used functionally. But the idea is maybe these Mueller cells, and there's some evidence to suggest this, are acting like fiber optics. You can see they're very large, almost look like phagocytes. And the theory is that perhaps they are, and there are many more of these than are shown here, perhaps they are serving to conduct the light from the anterior retina to the rods and cones in a manner similar to fiber optic structures. A fellow came up with this theory about 12 years ago, I guess. Because otherwise we don't have a very good explanation. But I'm going to come back. This isn't such a great illustration, so I'm going to come back and talk about phototransduction in just a moment. We've talked about the importance of the fovea. Make sure you know the difference between the fovea and the optic nerve, optic disc. We talked about this in the anatomy. We talked about the special properties of the fovea in the anatomy. We talked about macular degeneration. We said that a lot of things can go wrong here, but one of the things that happens in certain types of macular degeneration is that there is a destruction or deterioration of the neural layer in this region, and that tends to erode the patient's central or focal vision. The two general classes of photoreceptors are rods and cones. There are many more rods than cones. Rods are monochromatic, which means they're basically just sensitive to light intensity. Cones, on the other hand, are important for color vision and also for high discrimination, high visual discrimination.
What does that mean?
And if you look at all this is a sort of a relative abundance curve here, and what you'll note is that at the fovea, there's a much higher concentration of cones than rods, and then peripheral to the fovea, there's a high concentration of rods, diminishing as we move away from the fovea. Yeah.
Dr. King, what does it mean, what you just said? I'm sorry, you mentioned that the cones have high discrimination.
It means they are more essential for your ability. We talked about two-point discrimination in the skin. All right, your ability to resolve objects that are very close together. Cones are essential for your ability to resolve or neurologically separate, discern objects that are very close to one another. They are high resolution, like a high resolution monitor or a high resolution microscope. Your cones are more essential for high resolution imaging.
Okay, thank you.
On the other hand, having said that, if you looked out at the night sky and maybe you're watching the space lab, what is it called up there, the International Space Station, or maybe you're looking at a little blinking object, and you stare at it for a while. You're looking at it, and suddenly, like we saw in those optical illusions, it disappears. But you notice that while you're looking for it, you avert your eyes somewhat, it comes back. How do you explain that? You can go out tonight and try this. If you have a very faint star in your field of vision, if you look away from it a little bit, it will become brighter than if you look right at it. Why would that be?
Yeah, peripheral vision pick up light better.
Yeah, exactly right. Well, to a certain extent, and why, I guess is what I'm asking.
More rods.
The greater concentration of rods, which are basically just monochromatic light detectors. You have many more of those at the periphery of the fovea. Remember, the fovea, while it is good for it's good, it enhances resolution, it requires lots of light. You know, what happens at night to your color vision?
Goes down.
Tends to deteriorate because, once again, those cones depend upon the light for both resolution and color discrimination. I'm sure you in high school or college you did the little blind spot test where you take a piece of paper or an index card and you put two dots on it. You close one eye and you stare at one dot and then you rotate the index card, and what happens? That other dot disappears because at some point you're going to move it into the focal point of the blind spot, and you have no receptors there. So I don't think we need to worry too much about the infrastructure of these rods and cones. Just know that in general they have a segmented organization. Usually we talk about the outer segments and the inner segments here. There's going to be a cell nucleus down here. We'll talk about the pigments and the mechanism by which, we'll talk a little bit about the mechanism by which they operate and how phototransduction occurs in just a moment. But before doing that, I want to make sure, again, you appreciate the position of these rods and cones relative to this posterior epithelium layer. Those rods and cones, in effect, are buried to a large extent into the melanin, the melanin-related cellular processes, apical processes of those pigmented cells. The pigments in the various, especially cone receptors are sensitive to generally three different wavelengths. We can call them blue, green, or yellow. Experts use a slightly different terminology. But the key thing that I would just have you remember is that cones are not, well, they are able to detect colors because they have different sensitivities. Different cones have slightly different sensitivities in terms of the wavelength. You can see blue cones, red cones, green cones, and so on. Whereas rods don't really have that. They're most sensitive, as you can see, to sort of a green light, but they don't discriminate green. It's just light intensity is all that they really discriminate.
Okay.
Yeah.
So you're saying rods don't individually perceive blue, green, or yellow? There's individual blue rods, individual.
No, not rods, cones.
I'm sorry, cones. It's
Yeah.
individual blue cones. Okay.
Yeah, in different animals have different relative distributions, and that's why I don't want to get too much into this, because when we say blue sensitive and green sensitive and yellow sensitive, we're just using color, terms of color, which frankly we don't all agree on, to refer to wavelength. And so I don't know that that's all that essential. Just be aware, again, that cones are your color sensors. The rods, however, are much more sensitive. I think there's some evidence that under the right circumstances, one rod can sense one photon. Can you imagine? And there are all sorts. If you look this up, you can find that that's comparable to some seeing a flashlight. It's on the outside of the solar system. So I mean, it's an amazing capability of the eye. In any case, the rods are much more sensitive to light overall, but the cones are essential for color vision by virtue of their specialization. I want to talk a little bit about this process called phototransduction. it's described here, but instead of working through this, I'm just going to look at the picture on the next slide. But before doing that, what does transduction mean? We talked, not transfer so much, but something of energy. Remember we talked about this before, a transducer. Yeah, it converts. Yeah, so we're just converting information or energy from one form to another. And all of your sensors, to one degree or another, are transducers. They're converting energy of one type to usually action potential. That's where we're going to end up. So how does the retina convert light into these action potentials, with which we have come to appreciate as the basic language of our nervous systems? All right. So let's look at these little illustrations here. I think this will be a little clearer than sort of trying to work through that wording. So here's where this, on the left, we have a single rod in darkness. Up here is the pigmented epithelium. And so that yellow thing, all of this, would be the photoreceptor. So remember, the photoreceptor has its little head buried in the pigmented epithelium. And then in that membrane, it's not clear here, but in that membrane of the, I don't know if I have a picture back here, we're looking at, imagine one of these discs right here, the membrane of one of those discs that are present that make up the outer segment. So in that membrane is a specialized G protein. We give it a name, it's called transducin. It's doing transduction, we call it transducin, but really it's just a specialized G protein, and you already know how G proteins operate in general. And also in that membrane is a protein called rhodopsin. That rhodopsin, the rho implies sensitivity or relevance of red, but it's just rhodopsin. And that rhodopsin consists of two components. An opsin protein, a long chain of amino acids that we call opsin, and then this other component called retinal, which is very similar to vitamin A. So rhodopsin consists of opsin and retinal. All right, so under normal circumstances, when the rod is in darkness, you'll notice further along that receptor membrane, there are two channels. There's a sodium channel and there's a potassium channel. And both are leaking. Both are open. These are gated channels, but they're both allowing the ions to pass through. So we have both a sodium current and a potassium current when the eye is in darkness, when the eye is exposed to darkness. And so what is happening is that as both of these currents are available and present, there's going to be release of neurotransmitter from the photoreceptor to the next neuron, to the first neuron in the retina, usually a bipolar cell, but not always. So here's the deal. This is called the dark current. The dark current is simply the constitutive or tonic conduction of action potentials from the bipolar cell that are caused or induced by the release of neurotransmitter from the photoreceptor when the cell is in darkness. It's kind of baffling. But the darker the less light, the greater the current, the greater the release of neurotransmitter. Just sort of counterintuitive to what you would expect. Now watch what happens when we expose the photoreceptor to light. There's the light here. Remember, we have to put it down here because that's where the light is coming from, bizarrely. There's nothing logical about any of this. The light is coming in this way. It reacts with the rhodopsin protein such as that the retinal is released. Retinal is released, and now we're left with opsin. The opsin protein in the membrane changes conformation, resulting in a G protein cascade. The transducin changes conformation, and notice what happens here. What happens to the sodium current? It stops. All right, now you tell me what happens to the membrane potential if sodium current is, a sodium current that was present is no longer present.
Hyperpolarization.
Right. We go from a relatively high, a relative more positive membrane potential, again, I shouldn't use those terms, a more positive membrane potential to a more negative membrane potential because the potassium current is still there. And as the photoreceptor membrane hyperpolarizes in response to light, what happens to the release of neurotransmitter from the photoreceptor? It diminishes, yeah. There's always going to be, remember that word tonic, there's always going to be tonic release, and what we're doing is modulating. This is true for all the sensors, we're just changing the, modulating that tonic release, but it dramatically decreases. So here's the deal, as the photoreceptor responds increasingly to light stimulate, its communication with the bipolar neuron decreases. At least by that I mean there's less release of, there's diminished release of neurotransmitter. And so if you want to see in the dark again, if you want to have any type of visual discrimination in the dark again, what has to happen? Well, we have to bring the retinal back to the opsin. We have to have the restoration of rhodopsin. I think I mentioned a moment ago, retinal is chemically similar to what? Vitamin A. So you're going to have adequate dietary and metabolic vitamin A and its precursors, the retinoids, all those available in order for you, especially males, because males seem to have a genetic disadvantage here, to discriminate objects in the dark. I mean, how many times have you walked into a dark theater and you can't, you have no idea where you are until your eyes recover. By the way, this process is called bleaching. The bleaching refers to the loss of retinal with photoexposure. Yeah.
So is that why vitamin A deficiencies cause blindness?
Well, yeah, exactly right. With prolonged vitamin A deficiency, especially in children, you not only have this sporadic problem, but you have developmental failure of these circuits to develop properly, and that's irreversible. And of course, there are all kinds of transgenic, genetically engineered plants that have been proposed in areas where there is a chronic deficiency of vitamin A to try to compensate for that, to prevent. This is the most common, what you mentioned is the most common cause of childhood blindness in the world, a endemic or dietary insufficiency of vitamin A.
So are things bright to them all the time? Because like usually when I think of blindness, things would be dark because.
No, they wouldn't be able to sense, or they would. Vitamin A induced blindness, that developmentally induced is darkness. There would be no perception of light. So this isn't important. I just want you to understand that that as the photoreceptors are exposed to light, the bleaching process persists, and that if at any point thereafter one enters less light, one enters darkness, in order to have any type of visual discrimination, which you would need in normal circumstances, we talked about the wedding gown problem, for example. That retinal would have to be abundant and be available for reconstruction of rhodopsin, and that takes time. Photo adaptation, which we'll talk about just a little more detail in just a moment, takes time. Yes.
The degree of bleaching is, I see it says proportional to the amount of light. So it'll stay, if the light intensity does not change, the degree of bleaching will stay the same, independent of duration of exposure.
But it would be dependent on the duration.
Okay, so even if the intensity doesn't change,
Right.
The bleaching would increase.
Right. Yeah, both would be relevant in this context, both duration and bleaching. And again, there are other factors too. We indicated that as we age, we lose the recovery, we lose the ability to recover. Females seem to have a better capacity for recovery from bleaching or photoadaptation than males. I imagine there are probably, if you talk to an expert, there'll probably be 25 or 30 other factors that would contribute to this. And of course, you already know about this reflex. We didn't talk about the Edinger-Westphal nucleus and all that, but you know that this pathway is also part of the photo adaptation response. Yes?
Is there a certain amount or level of darkness that would cause, or a level of lightness that causes the retinal to separate from the opsin, I guess? Or
You're asking, is there a threshold at which the light
begins and ends?
Yeah, I don't know what that would be. I thought as you began your question, you were going to ask, is there a threshold of light at which it would be so large, it would be so great that that would induce irreversible damage to the photoreceptor? And I'm sure your parents told you not to look at the sun when you were a child. There is, but that usually, the way in which bright light destroys the photoreceptor is a little different than this. So the answer, I guess, to both questions is yes, but they use different mechanisms. But I don't know the threshold, the precise threshold at which you refer. So with respect to photo adaptation or light-dark adaptation, this is just a little summary that I would provide, although it's actually more complicated than that. The pupillary reflex that we just talked about, that would be one mechanism of light-dark adaptation. This is very rapid and is purely mediated by the parasympathetic nervous system. There is a neural component to photoadaptation we don't fully understand, but basically there are efferent signals that enable the brain to deal with persistent changes in intensity in light. For example, when you live in a cave on a regular basis, and if you think about it, to a large extent we do, as opposed to our ancestors. And then we've talked about the photochemical adaptation. This refers to the retinal and the bleaching and so on. When we talk about the photo adaptation, when your pupil becomes smaller, are you familiar with the concept of depth of field? We talked about pinpoint glasses and so on. And if any of you are photographers, are probably familiar with. But as your pupil becomes smaller and responding to bright light, your depth of field increases. And one of the problems with bright light is that you have a very shallow depth of field. Photographers use this for artistic effects and so on. But it's important to keep in mind, once again, that in bright light, depth of field diminishes. That's all we're talking about here, and we've already referred to the pupillary reflex. By depth of field, I mean you ever notice when you look at a movie, and your eyes are focused on the actress or the actor, if suddenly you get bored because you don't like what they're saying, you want to find something more interesting in the movie, you look around and everything's out of focus. Yeah, it's because of the controlled depth of field in those cameras. Same thing happens to your eye based upon the size of your pupil. Don't need to go through this again. I think because we've already talked about this in our discussion of the autonomic nervous system. What I want you to keep in mind, though, is make sure you know the two layers. Who was it? Bartek, I think, was asking about the iris. And I meant to bring in I have found a good picture of this. If you imagine the pupil, I don't know, maybe having the conformation of one of the rows here on the side of the room. Back there against the wall would be the smooth muscle. Again, simplifying this, would be these layers of smooth muscle. The sphincter muscle would be out here, and the dilator out here. But then anterior to that, up here, would be a variety of congeries of tissues involving that melanin that we talked about that's sort of embedded. So the iris is it's like an irregular mountain. It's not a very linear structure. We all have rather variable shapes to our irises. But in any case, you're right. This only refers to the posterior portion of the iris. And you can see the two layers of the muscle. Both of these respond to parasympathetic innervation. The inner muscle layer, the constrictor, also responds to sympathetic innervation. So if you look at our little illustration here, let's stop it. Go back. So the light enters the eye, and there it goes through. We talked about half of the information crossing over. By the way, as you look at this pathway, think about the neurological assessment you're going to be involved in later when you flash a light to a patient's eye and watch how why would you be looking for the response in both pupils? So you can see the stimulus, the action potential going back. All this is going back to the midbrain, regions around the midbrain. You can see a portion of this information back here. Oops, can't do that. Portion of information back here is crossing over as well. So you have crossing over anteriorly of the signals, and you also have crossing over, or partial crossing over at the level of the midbrain. And then there we're at the cerebral aqueduct. Well, that's called the Edinger-Westphal nucleus. I mentioned that earlier. And then watch what happens. Now when you have signals sent back from the brainstem to the ciliary ganglion, which you know to be a parasympathetic ganglion where there's a synapse. And then watch what happens when the signal gets to the iris there. Boom, it changes shape, becomes smaller to accommodate or to adjust to the increased light. You do not need to know the components of all the components of that pathway. I'm not going to ask you about the stuff in the midbrain. All right, any questions about the eye? All right, let's look briefly at the ear. As I say, I want to try to get through most of this for your test. All right, let's talk a little bit about sound. We talked a little bit about light. We talked about color and bass sensation and light. Sound is equally complicated, if not more so in some respects, at least with respect to the psychological presentation. We characterize sound in a variety of ways, but make sure, once again, you don't confuse the stimulus with the sensation. A sound stimulus is what? Can anybody explain to me what a, Steve heard this stupid line about if the tree falls in the woods and nobody's there to hear it, is there a sound? What is the stimulus that is responsible for your hearing a sound?
Vibration.
What is one big difference between vision and hearing? Vision, how many science fiction movies have you seen where the two spaceships are out there and engaged in battle and you hear boom, 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 right? And then the other spaceship blows up and there's this cataclysmic sound. Is that possible? No, why is that not possible? All right, there's no medium, there's no air, there's no conductive medium for sound in space, so there wouldn't be any sound. So it's important to understand that your hearing depends upon a medium. It can be liquid, it can be solid, but in general, we're just going to assume that it's gaseous, it's the atmosphere. So a sound, in terms of the stimulus, is simply anything that causes a vibration in the air. There's going to be compression of the air and rarefaction of the air. When I shut this door and the atmosphere is altered some way, is the alteration, I guess what I'm asking you, are the air molecules that are altered here, do they go into your ear? No. So so how is the sound propagated? How is it communicated from the door to your ear? Yeah, it's just a vibration back and forth of the air. And what's that doing in your ear? It's causing your tympanum, your eardrum, to respond and move back and forth sympathetically. So please understand that the stimulus is the excitation or compression and rarefaction of the air. What you make of that, like with the eye and the optical illusions, is a different story. But we call that the sensation. It's going to be different. So there can be stimuli, there can be excitations of different amplitude. We generally call those changes in loudness. Loudness is the psychological sensation. Sounds differ with respect to frequency. That's the physical term. Pitch is what we perceive, what we sense. Higher frequency, lower frequency, but you perceive that as a change in pitch. Although in fact, it's just a change in the rate at which the air is moving back and forth. Of course, duration is duration. This is perhaps the most mysterious one of all, and we don't really know if humans are unique in this regard. For example, if you were to sit down and talk with one of the lizards out there in the parking lot, maybe you've got to have your cell phone out and you play them a little bit of the Brandenburg Concerto that you were so fond of. would they hear it in the same way that you do? Not only they may not appreciate the music, they may prefer heavy metal or something, but it's very likely that they don't even hear it. They don't even hear the auditory stimuli in the same way that you do, by virtue of this concept of timbre. It's a French term called timbre. It's a funny spelling. It's not timbre. But this corresponds to waveform. Have you ever wondered, and we're not going to answer this right now, but how is that thing in your cell phone, that little diaphragm moving back and forth, able to communicate to you everything from a cricket chirp to a symphony? How is that possible? What can that little diaphragm do that is responsible for all those changes, that enable you, for example, to discriminate a trumpet from a saxophone? And if you think about it, your eardrum is doing the same thing. It's responding in the same way to this very limited medium of compression and rarefaction. In any case, your ability to discriminate qualities of sound is timbre. The quality of sound is called timbre, and that corresponds, without getting into a long explanation now, to the wave form of the auditory stimulus. So I just want to make sure when we talk about frequency, there's an inverse relationship between frequency and wavelength. We talked about this when we talked about the UV radiation. And by the way, the term hertz is used to describe the rate of vibration, and it's just 1 cycle per second, 1 vibration per second is a hertz. So if you read that normal human hearing is from 20 to 20,000 hertz, which I don't think is quite accurate. What does that mean? You hear vibrations from 20 vibrations per second to 20,000 vibrations per second. I don't know if this is working. Let's see how your hearing is. I don't think the speakers will handle all of these frequencies. There's one more above that. Did you hear it? I couldn't hear it. No. I'm not sure the speaker, and I don't think we can hear the lowest one either. We'll do it again just to annoy you. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9 of these. 1, 2, 3. I thought there was one more. But anyway, that gives you some idea. And of course, this isn't where we said, in theory, you should be able, your age, most of your age, you should be able to hear up around 20,000 hertz. They don't have CRTs anymore. Remember when you were a child, the television was a big screen, and that had what was called a flyback frequency. And when you turn your TV on, especially if it was a poorly engineered TV like the type I had, immediately you would hear a very high frequency that was on the order of the flyback frequency of that little electron beam going across the screen, usually on the order 16,000, 18,000 hertz. But in those days, as a child, I could hear it. Can't hear it now. In fact, I'm not sure. I'd have trouble hearing this. Of course, I'm not sure these speakers are a good test. Anybody familiar with brat busters? I sometimes use these in shopping malls and around 7-Eleven type stores. What are brat busters? What could they do around these types of establishments that would discourage young people and not affect the old people walking through like me?
It's a high-pitched sound that only the youth can hear.
Yeah.
It makes them want to leave.
Yeah. Ageism.
It's a high-pitched sound. People can hear, like, right outside stores. Anybody else
Most of the time, for a variety of reasons, we don't hear that full range from 20 to 20,000. Typically, most of us have a functional range from about 40 to maybe 14,000 hertz is typical, I think, for the average person functional. And then we talked about like vision, hearing declines with age, not merely in terms of frequency discrimination, but also in many other ways. One of the things you're going to find about the time that you begin to lose the ability to close focus is that although you can hear the TV set, you nevertheless need to hear it. You need to turn it up louder. And it's, yes, when you turn it up louder, is it annoying? Yes, but you have to do it anyway. Why? What's the problem with your hearing there? You can't discriminate the sound that you want to hear from the background noise. You lose the ability to focus on the sound. And this too is one of the major, it's called masking. One of the major problems with the presbycusis. Presbycusis involves loss of acuity and loss of discrimination. Where our ears are tuned, they're optimized to hear frequencies in the vocal range around, let's say, 3,000 hertz or a little lower than that. And of course, we don't hear as well as a lot of animals. Most animals have more acute hearing than we do. And of course, there's a lot of discussion about whether or not humans alone can respond to musical stimuli. And you may have seen this one on the internet or on Google. So you guys just want to dance with Snowball?
That is so cute.
All right. I love Snowball, but I can only handle so much of that. Our ability to respond to sounds, it follows a logarithmic scale. By that I mean that in order for us, how can I say this, that loud noises must be, they don't, very difficult to describe a logarithmic scale, but if you turn the volume control on your amplifier on your radio, and it says 0 to 100, I hope you realize that's not a linear relationship. 50 is not 50 times higher than 1, and 100 is not 50, or twice as big as 50. Rather, you are dramatically increasing on a logarithmic level the intensity of the sound coming from that speaker in your cell phone when you increase the volume, because of the way our ears respond to that. And the musical instruments are crafted in this fashion too. But we say that because hearing is logarithmic, the sound intensity is measured in what are called decibels, and decibels follow this logarithmic scale. So that typically, you can see some of the sounds here. Conversation is about 60 decibels. They're showing here a jet airplane starting up, about 125 decibels. So at that point, you run the risk of hearing loss, hearing damage. I want to talk a little bit about this timbre. We won't spend too much time on this, but remember, your ability to discriminate different types of sound depends upon the waveform. And this is what we're talking about by waveform, simply the way that the object vibrates. You can see some of that in this little demonstration. It's going to show us what that stroboscope of the guitar. So you can see how the strings are vibrating relative to one another, but that's the waveform of that string vibrating. So when the string vibrates along its length, it's also vibrating in the middle, in the thirds and quarters and so on. So that gives you some idea of that. But understand that when you see that vibrating string, it's not vibrating just along from the nut to the bridge of the guitar. It's vibrating in segments. And that the segmental vibration of that string is what you're converting into the musical sound of the guitar. It's magic. Let's watch this, then we'll take a break.
When a musician plays or
Let me
sings
get
A
the sound
or middle C, the sound source vibrates at 220 times per second. The sound source simultaneously vibrates in halves at twice that rate, 440 times per second.
Almost all objects do this.
The sound source also vibrates in thirds at a faster rate. And so on, in smaller and smaller fractions. Therefore, when a musician plays or sings a single note, numerous other frequencies are produced, but we perceive it as one sound.
He's just adding overtones there, and the quality of the sound changes. So this is sometimes called the chord of nature. When you sing, it's not just one tone you're singing all of these vibrations on top of that, and then how do I differentiate your voice from somebody else's or so on? It's because the relative strength of these overtones changes, and again, we call that timbre. Let's take a break and we'll pick up there.